Hello. We'd like to explain our recent paper about capybara genome. The title of our paper is The Dynamics, Causes and Impacts of Mammalian Evolutionary Rates Revealed by the Analysis of Capybara Draft Genome Sequences. Authors are only two, Dr. Isaac Barbarinde and myself. This paper was published in Genome Biology and Evolution in August 24th. You can see website and DOI, and this is the abstract. At this moment, as of September 14th, 2020, there's still one mistake in abstract. The genome size, expected genome size, is not 2.7, but 2.6, as shown here. Why capybara? 5 years ago, in 2015, Dr. Wenshan Lee, at that time director of Academia Sinica Biodiversity Research Center at Taipei, visited my laboratory in NIG, National Institute of Genetics. We discussed about evolutionary rate heterogeneity among mammals. Because body size was considered to be negatively correlated with the evolutionary rate of the species, we got interested in capybara that has the largest body size among the extant rodents. I remember Izu Shaboten Park, which is not far from our institute, has many capybara individuals. So I phoned. Then eventually we got some samples from recently dead capybara boy called Lai, as shown and below here, below photo. So we started genome sequencing. Okay, now Isaac, please start to explain this.
Yes, thank you so much, Saitou Sensei. So of course, the first thing that we were faced with was the assembly of the genome, because up to that moment, there was no genome for capybara. So we tried to assemble the genome, and we found that the genome size was about 2.62 gigabase pair. And then there are some other statistics about the genome of capybara. So the next question that we asked after assembling the genome, we tried to get the list of the genes, and we checked the closest species to capybara, which is guinea pig, as shown in the next figure. So in the next figure, we can see the capybara, which is Hydroqueris. So the next species to eat to capybara is caviar, which is guinea pig. So we tried to check if there was any differences in the evolutionary rates, and we found that more or less the evolutionary rates are so similar. So you can see in terms of the length, from the left, you can see caviar and capybara, they are pretty much similar. So then that led us to ask the report of Dr. Lee, that was already very old, 1986, where he reported that mouse and rats had very high evolutionary rates and human had very low evolutionary rates. So we try to ask whether that properties are representative of the respective orders, or do all rodents have high evolutionary rates, and do all primates have lower evolutionary rates? And as you can see in the phylogenetic tree, so when it is red, it means very high evolutionary rate. Then when it is deep blue, it means low evolutionary rate. And you can see it in the left and in the right figure, the B. So you can see primates which are blue, they tend to have lower evolutionary rates, and then rodents which are high, which are red, they tend to have higher evolutionary rates. So these are some of the things that we found in the studies. So it's interesting that although this study was done when, Dr. Lee's study was done when there were fewer species, we found that mouse and rats actually have the highest rate, and humans have very low rates. So the next question, of course we were able to confirm also the previously reported slowdown of evolutionary rates in hominoid common ancestor, and we found in addition that there was an acceleration of evolutionary rates in the common ancestor of mouse and rats and other species. So this acceleration of evolutionary rates may be responsible for increase in species. Actually, the descendant of that particular acceleration, that is Muridae. Muridae has the highest number of species among all families in mammals. So this acceleration of rate might be responsible for increase in the number of species. So these are the dynamics that we found. Then we try to find what could be responsible for differences in evolutionary rates. So we try to investigate some previously reported factors, and you can see when they are red, it means that they are positively correlated. When they are blue, they are negatively correlated. We found a number of factors that are actually correlated with the evolutionary rate, but we try to see which ones can actually predict the evolutionary rate better. And we found that the three most important factors are the taxonomic orders, that is the order to which the species belong to, the generation interval, and the litter size. So these are some of the things that have been reported, especially by Ota Sensei of National Institute of Genetics. So the next thing that we try to investigate is the impact of this rate heterogeneity or difference in evolutionary rates. We look at the transcriptome data, and we actually found that the transcription pattern is similar. We found higher rates in rodents, and we found lower rates among primates. So this pattern is closely matched with the evolutionary rate pattern that we previously shown. So this, of course, we also hypothesize that the increase in the number of species that we found in Meridian family might be related to the increase in evolutionary rates. So these are some of the consequences of difference in evolutionary rates dynamics that we report. So this is an interesting study, but there are still some unanswered questions, and maybe one question that everybody would like to know is which part of the changes are associated with body size. So we could not address this very extensively because of the limited coverage of our sequencing data. But now there are at least maybe 2 other efforts to sequence the genome of capybara. Hopefully we can include this analysis in future studies to see what actual genomic regions are associated with differences in body size. Thank you.
Mute, please. Okay. Yeah, thank you. So I'd like to add something. So when Dr. Babarinde extracted DNA, I asked Professor Itsuro Inoue's lab, Human Genetics Laboratory in our institute, to sequence because they have a next generation sequencer called MySeq. And we just used some small kit. So essentially, genome sequencing was cheap. We don't need to ask some huge sequencing center. Now genome sequencing is cheap. The most important part is analysis. This is what done by Dr. Babarinde. So anyway, as he mentioned, heterogeneity is very interesting, but still there are so many mysteries. We have to have many questions already mentioned by him, but also still we have to explore the genome. Thank you for your hearing.